Welcome all to our first contributory talk session. And before introducing the speaker, let me remind you a couple of things. To make questions, please remember to use the question and answer button. Do not raise hands. Do not use the feed tab. And remember also that you can vote a question made by other attendees, and then the platform will automatically rank them according to the number of likes. So this is useful for the session chairs and for us to prioritize questions during the discussion, so please do that. Okay, so let me introduce our next speaker, SimoneTiberi from the Institute of Molecular Life Science at University of Zurich. He's going to present his work with the title Distinct, a novel approach to differential distribution analysis. So Simone, if you're ready,
okay,
you can start.
I'll get started. So thank you for the introduction. I am SimoneTiberi. I am a postdoc at the University of Zurich. As Chiara said, I work with Mark Robinson in basically developing statistical methods for bioinformatics, because I originally am a statistician. And Distinct is our second bioconductor package, our second tool in the field. So Distinct is a very general method that allows you to do differential analysis between groups of distributions. And although it's very general, it's ideal to applications on single-cell data, like single-cell RNA-seq or flow or mass cytometry. And on these types of data, you can, for instance, perform differential state analysis, which are differential analysis on subpopulations of cells. Now, canonical tools for differential abundance tend to focus on shifts in the overall mean. And so they fail to capture, or they're not ideal to capture, variations that single-cell data can show. For instance, when some cells are stimulated, and as a consequence, some are upregulated while others are downregulated, but the overall mean between conditions is similar. Distinct instead compares full distributions, and so it can identify the changes regardless of whether they involve the mean or not. Implementation-wise, as I said, it's on bioconductor. So now I'm going to illustrate the methodological and, in general, conceptual aspect of the method, and then I'll show you how it performs in simulated and experimental data sets. So the key idea of the method is to look at the ECDF, so at the empirical cumulative distribution function of the data. And this allows you to discern complex variations between distributions. So if you take these differential patterns that were proposed by Kothauer for single-cell RNA-seq data, you have canonical differential expression, where there is a shift of the entire distribution, differential proportion, dP, where you basically have two bimodal distributions that are a mixture of two distributions, but with a different proportion of the two mixtures. Then you have differential modality, which compares a unimodal and a bimodal distribution, and dB, which, like the M, compares a unimodal and a bimodal distribution, but in this case, the overall mean is the same under the two conditions. So if you look at the CDF of each one of these profiles, you will be able to discern the two densities. Obviously, in DE, that implies a complete shift of the distribution, while, for instance, in dB, there will be an overlap in the center around the mean and the median, but there will be differences in other areas. So if we take dP and more realistically look at an example with a biological replicate, so we introduce sample-to-sample variability, what we would like to do ideally is to numerically, mathematically quantify this difference we see between the CDFs. And the way we do it in Distinct is basically to build a fine grid with vertical cutoffs, and then at each one of these cutoffs, we compute the absolute difference between the mean ECDF in a group and the mean ECDF in another group. And we do that for every cutoff and simply add these absolute differences. And this leads us to a test statistic S, OBS, because it's the one observed on the actual data, that somehow should summarize how different the groups of distributions are. Now, in order to translate this number into something like a p-value that allows us to perform differential testing, we cannot rely on asymptotic theory, because there is no asymptotic theory telling us how S behaves under the null. So to do that, we use a non-parametric permutation approach, where instead of permuting the samples, which is typically not possible because on biological data there are not enough samples, we permute the individual observations. So in this case, the single cell measurements. And this is normally possible because there are many more cells than samples. Now, it's important to note that in principle, this may actually lead to an inflation of false positives because there is a lack of exchangeability. But we benchmark this thing in simulated and real data sets, and in all cases, it has good control of false positive and false discovery rates. Then, once you have done your you computed your permuted statistics for your p permutations. Computing a p-value is trivially obtained by counting how often your permuted statistic SP is greater than the observed statistic. Now, if nuisance covariates like batch effects are available, then we simply feed a linear model and we try to regress them out. So we feed a linear model where, as a response, y, you have your input data, so your CPMs, log CPMs, whatever you provide, you feed in this thing. And then on the other side of the linear model, you obviously have an intercept, and then a covariate effect and some residuals. Now, note that these covariates are nuisance covariates, so things you want to get rid of. We're not talking about the group comparison you're interested in. So what this thing does, if you provide covariates, it will simply fit this linear model, estimate via least square regression the parameters of the model, and remove the effect of covariates from your input data. And so then it will perform differential analysis exactly as I showed before, but on these normalized, regressed values. Keep in mind that if you do this for differential state analysis, as I said, you do differential testing in populations of cells. And so we actually allow for the effect of covariates to vary between genes, obviously, but also between clusters. And we fit, therefore, this model for every gene and for every cluster separately. So to briefly wrap up the conceptual details of the method, since we use a non-parametric permutation approach, we don't rely on asymptotic theory, which could be inaccurate when you have few samples. And we don't rely on any specific parametric model, which is actually quite hard to formulate accurately for single-cell data. And importantly, since we don't have any assumptions on how the data was generated, the method is very flexible. And so it can be applied to various types of data, even beyond the single-cell data applications that we consider. The method also allows for biological replicates and can adjust for covariates. And importantly, since we have no assumptions on the model, you have to normalize data in advance. So you should provide CPMs, log CPMs, and not row counts. So getting into the simulation studies, we first consider a differential state simulation study on mass cytometry data, which comes from this site, where spike-ignited signals were computationally introduced in experimental data. And so this has all the properties of real data, but also embeds a ground truth signal. And we compared this thing to two methods from this site based on lemma and mixed models. And we chose them because on this exact data set, they outperformed the competitors. And we considered, still from the original paper from this site, 3 null datasets and 3 non-null, what I call discovery datasets. So the main one and 2 more where the effects, the size of the differential effect was diluted by 50 and 75%, so making it basically harder to detect differences. So in this plot here on the top, we have the true positive false discovery rate for the non-null dataset. And then at the bottom, we have the densities of the raw p-values under the null datasets. So let's start from the top first. This thing is in red, while diff side methods are in green and blue. So what you can see is that in the main simulation, all methods are pretty much equivalent. But then as the size of the differential effect decreases, there is a significant difference in true positive rate between methods, which shows that this thing actually has good statistical power in detecting effects, even when they are relatively small in magnitude. And at the same time, it also controls for the false discovery rate, as you see from the from the circles that denote the observed false discovery rate. And from the bottom plot, you simply see that all methods have roughly uniform p-values in the null semi-simulated data. We then considered a second simulation, actually a bigger simulation from Muscat, which is a biconductor package from Helena that allows you to simulate droplet single-cell RNA-seq. And again, we consider a null and a non-null simulation. And here, interestingly, we could simulate differential profiles from each one of the 4 differential patterns that I showed you before. And again, we compare this thing to mixed models and to pseudo-bulk approaches from Muscat. Data, when needed, were normalized in canonical ways, so CPMs, log2 CPMs, and residuals from SC transforms normalization. Again, we have true positive force discovery rate. Here it gets a little bit complex to visualize fully because you have one for every differential type separately. So this thing, according to the input data, is in red, EdgeR in blue, Lemma is in green, and mixed models in gray. What you can see, if you like, the same plot is also in the preprint if you want to zoom in and visualize it more carefully. You can see that all 3, the results from this thing on all 3 input data and from 2 pseudo-bulk approaches, in particular, one from EdgeR and one from Lemma trend, are very similar in differential, canonical differential expression. They're pretty much tied together in DP here and in DM here at the top. But then there is a significant gain in the true positive rate from distinct in DB, which, if you remember, is the differential profile where there is no change in the mean between conditions. And in all cases, the false discovery rate is well controlled. And so this shows us that when there is a difference in the mean between conditions, this thing performs similarly to pseudo-bulk methods. But then it provides an advantage when there is no difference in the mean, which is expected from a conceptual point of view, because instead of looking at the mean parameter, it compares full distributions. Also interesting to note that if you look at mixed models, which are in gray here, they have fairly low statistical power, so we excluded them from the next analysis. These are the results from the null simulated data. Again, p-values are fairly uniform for all methods, even conservative from some. And from a computational point of view, here we have the time in minutes that each method required to run on a single Muscat dataset. Now, these are fairly small datasets because they only have 4,000 genes and 3,600 cells. You see that pseudo-bulk methods only took very few seconds, while this thing needed between 4 and 5 minutes, which I'm definitely biased here because I'm the author of the package, but it seems a very reasonable time to me, because this thing has to compute a permutation test for every gene cluster combination. So here we're talking about 12,000 permutation tests. And then time increases a lot for mixed models, and it goes, it ranges between 30 and 300 minutes. Keep in mind that these were methods use 3 cores for distinct and mixed models, while pseudo-bulk actually only run on a single core because they don't allow for parallelization. Okay, getting into the experimental data sets. We firstly wanted to investigate false positive rates, and so we considered 2 null experimental data analysis where no differential signal was expected in the data. And so we consider first a data set that I call T cells. That's a SMART-seq2 data set of 11,000 cells from 12 colorectal cancer patients. And a second data set that I call TCANG, which is a 10x droplet single-cell data set from 8 lupus patients that were observed before and after treatment. Obviously, in the null analysis here, we only looked at the control samples. And to be accurate, we also removed one of the 8 because it looked like an outlier. And in both datasets, we randomly separated the samples in 2 groups 3 times so that we generated 3 artificial replicate datasets. And here are the raw p-values from the methods. And going from the most conservative to the least conservative, you have Ejar and Lim-Mavu, which appear to have very conservative, uniform, or even inflated towards 1 p-values. This thing has a marginal inflation of false positives in the Kang dataset, which decreases in the T cells, leading to almost uniform p-values, while Lima-Trend has a significant increase of false positives in both. And lastly, we took again the Kang dataset, and now we perform this time a differential analysis looking for actual patterns between controlled and stimulated samples. And we found firstly that this thing identifies more differential patterns than pseudo-bulk methods, as expected. And we also found several, well, according to my judgment, interesting non-canonical differential patterns that were identified by this thing on all 3 input data, and that were not detected by any of the 5 pseudo-bulk approaches I showed. And here are some of these profiles that I selected. So these are densities at the group level. So we're aggregating the sample level counts and plotting them at the group level. So these patterns mostly look like a DP profile, where one of the 2 distributions is typically 0 or like low values, and the other one is non-zero or still higher density, higher values. You might be thinking that these are not crystal clear patterns, but remember that DP, when is strong enough, also implies a change in the mean. And so these are very selected, very filtered patterns because they're not detected by any pseudo-bulk methods. So clearly that excludes a lot of interesting DP-like profiles where there is a significant change in the mean. Now, I think doubtlessly the most interesting one is this one on the bottom left, that resemble like a DB pattern where you have a roughly unimodal distribution for controls and bimodal on the sides for the stimulated samples. And if we zoom in and look at the actual sample level densities of this pattern, so here you have the sample level densities and CDFs, and then at the bottom the group level density and group level CDF. You can see, or at least I want to highlight 2 things. First is that this is, I think, a very clear pattern. So if you look here, you see that all the red curves, which are controls, are above the blue lines in the center and below on both sides. And the second thing I want to highlight is that, as we expected and we hoped, this thing is able to capture and identify this pattern even if the ECDFs cross roughly in the middle of the distribution, and obviously multiple times for the sample level ECDFs. Similarly, in a different differential profile, which is less clear than the previous one, I want to highlight that, again, the method is able to capture and identify this profile even if for the majority of the plot and so of the cutoffs and the grid that we build, the ECDF curves are pretty much overlapping between methods, both obviously at the group level and at the sample individual level. But this thing is still able to identify because they differ in a subpart of the entire plot. So to briefly wrap up results, we have developed Distinct, which is a general method to compare groups of distributions by looking at their ECDFs. The method targets complex changes regardless of whether they involve the mean. It allows for biological replicates and can adjust for covariates like batch effects if present. And in my opinion, at least, it has good performance in simulated and experimental data. And again, since it's fully non-parametric, it has a permutation approach. It avoids parametric assumptions. It doesn't need asymptotic theory, and is very flexible. Again, it can be applied also in other types of data and other contexts beyond the ones that we consider. As I said, it's on Bioconductor, and there is a preprint if you're interested. And finally, I'd like to thank those who contributed to the work, and in particular Helena, Lucas, Pandelis, and Mark, and in general, the Robinson Lab, and in particular Almut, for their feedback and contributions. And this is a pre-COVID time picture of the lab. So thank you for your attention, and I'm happy to take questions.
Okay, thank you, Simone, for the nice talk and for the interesting approach. There are a bunch of questions, starting from the most upvoted. A question from Levi. Your tests seem related to the Kolmogorov-Smirnov test. How do you compare with it?
So the Kolmogorov-Smirnov, right, is quite different. One big difference is that in that one, you only compare two distributions. So this is basically an attempt to extend those kind of tests, like those kind of full distribution tests that compare two individual distributions, to a case where you actually compare groups of distributions. So you cannot apply, the main difference is that you cannot apply Kolmogorov's test if you have groups of distributions. I also think it doesn't provide a p-value and just a ranking, but that's, I think, a secondary issue.
Okay. Okay, then another question from Nils Kurzawa. Very cool method, nice talk. What is the minimal number of cells per group one needs so that your method is applicable? And I think that there should be another one related to that regarding the minimal number of groups one needs so that your method is applicable. So the number of replicates and number of groups.
Number of cells. Did they mention where? Okay, never mind. Groups, you need two groups. So it performs like a two-group comparison. Replicates, we didn't investigate these, but technically, I mean, with two replicates per group, you should be able to, I mean, it can work. And interestingly, cells. Ideally, even with few cells, you have enough to apply a permutation test. For what concerns actual performance, detecting effects, I guess like with any other method, if you have very few cells, then obviously you will have very low statistical power in detecting those effects. We have benchmarked up to, I mean, we have done several simulations where we have 50 cells per group in each cluster of cells. So 50 cells per group in each in each cluster, and the methods work, but obviously they had like fairly low true positive rate, but that was true for all methods because the size of the differential effect was very small, and all methods work better with 100 cells per sample in every cluster. But again, it depends on like how strong also the effect is. I think that's more of a general question for all methods.
Okay. Another question from Davide Risso. What do you think is the reason for the low performance of mixed effect model in your simulation?
I haven't thought about it, honestly. I added them recently before the final submission. I didn't investigate or think too much about it.
Okay.
There is maybe also the Muscat paper from Helena. I'm not sure. I think they might show the same attitude there. We have different simulations in the Muscat paper that was just published, but I think they also have a similar performance there. I'm not sure.
Another question from Milan Malfait. Do you have an idea why the exchangeability does not seem to play a big role for the FDR control? And did you benchmark this on different data sets?
Well, we did benchmark, we did consider several data sets. We have, we did quite a lot of, we have two different scenarios for the simulations, mass cytometry and our single-cell RNA-seq. And in both cases, we have the non-null and the null, and then we have like two null data sets. So we did quite a few investigations on how the false positive rate and the FDR behave, and they seem to behave fine. Why does it not play a big role? I think because you need a very high sample-to-sample variability in order to have an inflation of false positives. I think when the variability between samples is very, very, very big, much bigger than the effect you have between groups, that then might play a role, but that's typically not the case.
Okay. I do not see any other questions. I think we have answered to most of them. And okay, we are perfectly on time. And now there is a break of 10 minutes, and we will be back for the next section with the keynote. Thank you, Simone. And
thank you all for listening.
Yeah.